Here I present a paper about the role of serotonin in the early development of animals. I chose this paper because it merges the two themes of the endocrine system, where serotonin is a major player, and development. The central question of this paper is, how does the body organization of a multicellular organism arise? So different organismal groups have different body symmetries. For example, there are spherical volvox algae, radial, for example, the sea anemones, chiral, which snails are, bilateral, for example, these planaria worms, and pseudo-bilateral, which is, for example, us vertebrates. And pseudo-bilateral means that they generally have a bilateral symmetry, but a few things are asymmetrical. For example, we have paired eyes, limbs, kidneys, nipples, and so on, but we only have one of each: heart, liver, spleen, gut, and so on, and they all are somehow stuck in the body without any symmetry. So we'll focus here on the question of what tells the body, when it is in development, where left and right are to make these organs, or the asymmetric development of paired organs such as the brain hemispheres and the lungs. And the answer is not due to randomness, because these things are almost always on the same side between individuals. Like, the heart is always on the right side. Although sometimes the laterality is switched, which is called situs inversus, which affects about 1 in 10,000 humans. They don't have the heart in the right place, so to speak, but are otherwise usually perfectly healthy. This paper now is looking at the role of a likely candidate, which is serotonin. Serotonin is widely distributed in the body and has loads of different functions. In the brain, for example, it affects depression, sleep, aggression, food intake, psychosis, anxiety, and many others. You might have heard of it as a happiness hormone, maybe. Lesser known are its effects outside the brain, where it is involved in regulating blood pressure and volume, cell regeneration, heart functions, intestinal movements, the immune system, organ development, and others. By the way, foods with higher ratios of tryptophan, which is an essential amino acid, to other amino acids, might actually lead to more serotonin in the brain. So it might make you a little happier. And that also makes sense, because tryptophan is a precursor for serotonin. There is a hypothesis that many domesticated crops, like these chickpeas, have maybe unknowingly been selected for higher tryptophan ratios. And similarly, treating corn with akali also increases the bioavailability of tryptophan, which is why making hominy might have become a tradition in some cultures. To go on yet another tangent, serotonin is also called 5-HT in the literature, and you'll also see that in the results later. And this stands for 5-hydroxytryptamine, as there is a hydroxy group at the fifth carbon atom of the tryptamine molecule, which makes this serotonin. But back to the paper. Serotonin is a good candidate because of its known role in organ development, and also because most cells, and not just neurons, can make and sense it. So the specific question is, what role does serotonin play in invariant left-right asymmetry in otherwise symmetrical animals? This is partially exploratory research, so there are many sub-questions and sub-hypotheses attached to that we'll go over. Now to the methods. They used Xenopus laevis, which is the African clawed frog, for the study because this is a very well understood model organism for developmental biology, and that means that they had all the molecular tools to work with it. The researchers took some eggs and developing blastocysts in different stages, and they exposed them to different chemicals which inhibit different types of serotonin receptors. There are 7 receptor types to which serotonin can bind, and they all make different things happen once they are activated, as you can see here in this table. Most are slow, acting through the G protein CAM pathway, and one is a fast-acting ion channel. Most are excitatory, but 2 are also inhibitory. So that shows you that it's by far not enough to know that a molecule and a receptor are present to know how it will affect the cell or organs or even the whole body. The researchers also did the opposite, which is upregulating the levels of serotonin, but not by adding serotonin itself, because that usually doesn't work. But in the cells, rather, serotonin is broken down by monoamine oxidase. The researchers then just inhibited this enzyme, and that means that more serotonin is floating around in the cell and not being broken down. They then let the embryos develop a bit and scored on which side the organs were forming. So the most basic question is, does serotonin play a role at all? And the result is yes. Specifically, inhibiting the R3 and R4 receptor types makes some organs switch to the unusual side, which is also called heterotaxia. Also, increasing the serotonin levels leads to the same outcome, which means that the cells probably need a certain level of serotonin to function well. The next question is serotonin produced in the embryo, or does it come from the mother? When the team added a chemical which inhibits the synthesis or production of serotonin within the cell, it didn't do anything to the asymmetry of the organism. So the serotonin probably comes from the mother and is not synthesized in the cells. Next question. When does the lateralization happen? When we compare the outcomes of manipulations in the different cell stages and looking at the serotonin concentrations over time, we can conclude that serotonin is probably most active from the 4-cell stage until stage 7, so to the end of the cleavage phase. So now we know when, but where exactly does serotonin affect the embryo? The scientists injected the chemicals into each of the cells separately and saw the greatest effect of messed up asymmetry with the right ventral cell. So that's probably where most of the stuff is happening. But maybe the biggest question is how serotonin interacts with the cell in this early development. This study investigates 3 options: gap junctions, ATPase, and microtubules. Gap junctions allow for a rapid exchange of ions or information between neighboring cells, which is rarely the case in the body. And ATPases, as the name suggests, create ATP, which is an energy storage molecule, and they do this from differences in the concentrations of certain ions between membranes or cells. Microtubules give the cell structure, and they are used for the transport of molecules within the cell, so they're also likely candidates. Blocking each of these cell parts individually reveals that both gap junctions and ATPase are involved, but not microtubules. So that suggests that it is mostly the flow of ions within and between cells that is important. And the last question this paper answers is what downstream genes serotonin affects in the right ventral cell. And the answer is at least sonic hedgehog, which is a gene that is well known for its important role in organ development, and especially the development of extremities. I know that this was a lot, so here again are the main conclusions of this paper. The left-right asymmetry arises from a higher concentration of maternal serotonin in the right ventral cell of the 4-cell stage. Serotonin mainly affects ion flows between the cells, but also some genes indirectly through cascades. And this in turn changes expression patterns of downstream genes that are known for regulating body organization. But this is, of course, not the whole story. And in particular, other studies have shown that serotonin transporters are also involved. And the flow of the water and stuff outside of the cells, or the developing embryo, through cilia, is also involved in the asymmetry formation. And there is a huge debate now over whether this flow around the cells is enough to create asymmetry, or whether it's just a downstream or side effect of the different ion flows between and within the cells, which are also mediated through serotonin, as we saw and explored in this paper. Another open question is what makes the serotonin itself be distributed asymmetrically between the cells or within the zygote? So that was my deep dive into molecular developmental biology. I hope I haven't overwhelmed you too much with this information and rather left you wanting to learn more about this fascinating machinery that makes an organism develop and function. Now, stay happy and boost your serotonin with some harmony, sunlight if you have it available right now, some exercise, or being social.